In this how-to guide presentation, I will take you through step-by-step on how to use the TARGATT knock-in kits and the TARGATT technology to generate site-specific transgenic mouse models. Now first, what is TARGATT? TARGATT is an integrase-based site-specific gene knock-in technology. Integrase recognizes and recombines between two sequences, ATTP and ATTB. Upon recombination between ATTP and ATTB, it results in an integration of a gene of interest, and at the same time, it creates two hybrid sites, ATTL and ATTR. These two new sites can no longer be recognized by integrase. Therefore, integrase catalyzes an irreversible and unidirectional reaction, increasing site-specific gene integration efficiency. Now, how can we use TARGATT to generate site-specific transgenic animal? The general process is basically the same as the conventional method in generating random transgenic animals via pronuclear microinjection. So basically, one-cell embryos are collected from embryo donors. In this case, these embryo donors have ATTP site already engineered in their genome. And these ATTP containing embryos can then be microinjected with the target vector that contains gene of interest, plus the integrase mRNA. The injected embryos can then be transplanted into recipient pseudopregnant mothers to carry the embryos to term. Pups will be born and then genotyped for potential transgenic founders. Now, this how-to guide is consisted of 3 parts or 3 steps. So part 1 is on how to design and clone the target vector. Part 2 is on how to obtain target mice and collect embryos for pronuclear microinjection. And part 3 is to identify founders by genotyping. Now, the first part is to clone the target vector. I just want to mention that we have a collection of TARGATT starting plasmids that contain various promoters, reporters, and lock-stop-locks P cassette, depending on the purpose of the project. So in general, the construction of the target vector takes 1 step cloning. So gene of interest can be made either from another vector, or it can be amplified by PCR amplification, or it can be synthesized. This fragment can then be subcloned into a target vector that has multiple cloning sites for the ease of cloning, and also contains ATTB site for integrase-mediated recombination. And this subcloning step will generate the target vector that containing gene of interest. So the next step is to prepare and purify the target or plasmid DNA for microinjection. The quality of the target vector or plasmid DNA is very crucial to the success of using the TARGATT technology for generating site-specific transgenic animals. So first, the DNA can be prepared using MIDI-prep kit to extract plasmid DNA, and purification can be done using column purification method. And for quality control of the plasmid DNA, we use several criteria. One is to determine the concentration accurately. This is important because the concentration of the DNA that is injected into the pronuclei of the embryos is usually very low. So the accuracy of the concentration is crucial. We also use restriction digestion to determine if the plasmid is correct using fragment sizes after restriction digestion. In some cases, we also do sequencing to confirm that the DNA is all correct. It is also important to test for absence of RNase. This is because the integrase that's microinjected into the embryos is in the form of mRNA. So it is crucial that the DNA prep, or in that case, any other reagents, do not contain any RNase. We also test for the form of the DNA in the prep, because only the supercoiled form is effective in doing gene integration. So our cutoff percentage is 70%. So we require that 70% of the DNA is in supercoiled form in a prep in order to pass our QC standard. So this agarose gel shows an example of a DNA prep. So usually there are three forms of the DNA in a prep: open circle, linear DNA, and supercoiled. And so the supercoiled DNA, which usually runs as the fastest band on the agarose gel, is a good form for recombination. So 70% is, like I said, a cutoff percentage in order to pass the quality of a plasmid DNA to move on to the next step, which is microinjection. So for microinjection, we make a mix of the DNA with the integrase mRNA. And the final concentration is about 3 to 5 nanograms for the DNA and 50 nanograms of the integrase mRNA. And the mixture is microinjected into the one-cell embryo. And one important note here is that we usually only keep the plasmid prep and inject within 2 months post-preparation. Now, part 2 is to get target mice for microinjection. So we have H11, C57 black 6 strain ready for purchase directly from us. And these animals are shipped directly from Charles River Laboratories. They shipped internationally anywhere in the world from Charles River Laboratories. And for other strains, including C57 Black 6, Rosa 26 Locust, FEB Rosa 26, and FEB H11 strains, please contact us first and we'll make arrangements. So how to obtain one-cell embryos for microinjection? So this is pretty similar, like I said, as the process for generating random transgenics. So first, buy target animals, and then usually the animals that shipped from Charles River are female target animals, and these animals can be bred with wild-type males, and then one-cell embryos can be collected from the donor, and then these embryos can be used for microinjection. So effort should be made to microinject the female pronucleus, since the female contain the target ATTP site. In a case where female pronucleus are not easily identifiable, an effort should be made to inject both the male and the female pronuclei. And the last part, which is to identify founders. So we have genotyping kits, which will allow identification of founders either at the H11 locus or at the ROSA 26 locus. So basically, microinjection is performed, and then embryos are transferred into pseudopregnant females, and pups are born. And then these pups are genotyped using a PCR strategy. So we usually cut about 2 millimeter tail snips, and then this is placed into lysis buffer and using proteinase K. And then the genomic DNA is used for PCR reaction. And then in the genotyping kit, we have set primers for PCR and also protocols to identify potential site-specific transgenic founders. So this concludes our TARGATT short guide on using the TARGATT technology to generate site-specific transgenic animals. I'd like to thank you very much for your attention. If you have any questions, please contact us at 408-773-8007 or email us at info@appliedstemcell.com. Thank you very much for your attention.